Hello everyone and welcome to the Academy of Eating Disorders webinar titled Genetics in Eating Disorders with speaker Professor Tracy Wade of Flinders University. My name is Emily Matheson and I have the privilege of completing my PhD in clinical psychology under the supervision of Professor Wade. I'll be your moderator for this evening, and before I go on to introduce you to our presenter, I'd like to run through a couple of housekeeping details. Firstly, there will be a 30-minute question and answer session at the end of this presentation, and we invite you and encourage you to submit your questions as we go along through the presentation, using the questions feature on our webinar control panel. Questions submitted through the webinar will be addressed at the end of the presentation. Please remember to include your name and location along with your question. Second, if you experience any technical difficulties while participating in the webinar, please use the questions feature to contact staff. AED Director of Communications, Etta Carter, will be standing by to help you out. Now I'd like to move forward and introduce you to our presenter for today's webinar, Professor Tracy Wade. Tracy is a professor in the School of Psychology at Flinders University and is currently the academic lead on the Management Committee for the statewide Eating Disorder Service in South Australia. She is also a member of the Steering Committee of the National Eating Disorder Collaboration here in Australia. With over 30 years of clinical experience, Professor Wade has had a profound impact on the eating disorder community, both with respect to her clinical engagement with patients as well as her extensive research. Professor Wade's main research interest is in the area of eating disorders, in particular the etiology, genetic and environmental risk factors, the prevention and treatment of eating disorders, as well as the impact of perfection. She's been the Chief Investigator for several NHMR grants and is currently the Chief Investigator for an NHMR funded project relating to online eating disorder treatments in young adult women. Over the course of her research career, Professor Wade has provided us with over 150 peer-reviewed publications, including books, book chapters, and journal articles. In addition to her publications, Professor Wade has shared her wealth of knowledge in national and international conferences and workshops around the world, spanning from rural Australia to London, England. Professor Wade's excellence in research has not gone unnoticed. In the year 2000, Professor Wade was the recipient of the Ian M. Campbell Memorial Prize for outstanding contribution to the scientific or professional status of clinical psychology in Australia. And in 2003, she was awarded the Australian Psychological Society Early Career Award. And only recently, Professor Wade was again acknowledged for her ongoing contributions and was elected a Fellow of the Academy of the Social Sciences in Australia. In her spare time, she enjoys walking, gardening, embroidery, cooking, reading, and spending time with friends and family. It is my absolute pleasure to now hand you over to Professor Tracy Wade, where she will discuss genetics and eating disorders.
Welcome everybody, and thank you for joining us. I know that it's slightly challenging with all the different time zones, so thank you very much for getting out of bed or keeping away from bed in order to join in and participate. I'm talking about the genetics of eating disorders today, and the first point that we've known for quite a long time is that eating disorders run in families. So we know that there's a 7 to 12-fold increase in the prevalence of anorexia nervosa or bulimia nervosa in relatives of people who have eating disorders compared to families where there are not eating disorders. Now, we've known this for quite a long time, but the why is something that has remained a bit more elusive. So, in the very early days, and we're talking about the 1800s, our earliest focus was on the and there was a belief that negative influences within the family must cause eating disorders. This is why you see clustering of eating disorders in families. And Charcot talked about the family being a pernicious influence, and Gall talked about them being the worst attendants. And this tradition was carried on. We have, like most areas of psychiatry, a strong tradition of hypothesizing that the family had caused eating disorders. So most of us would be aware of the work of Salvador Minuchin and his work on the psychosomatic family model. And he suggested that a prerequisite for the development of anorexia nervosa specifically was a family process that was characterized by rigidity, enmeshment, over-involvement, and conflict avoidance, which occurs alongside a physiological vulnerability in the child and the child's role as a go-between in cross-generational alliances. So this belief led to some early approaches in family therapy, and the belief about the involvement of the family still continues to this day, despite the fact that we now have a lot of evidence to show that the reason that eating disorders run in families is because of genes. So I've just pulled out this quote from 2015 paper. It was in a family therapy journal, and it says, the decoding of the human genome has made possible research on the genetics of anorexia. This has failed to confirm the expected relationship. A number of leaders in the field of family therapy advocate the idea that family relationships do not cause anorexia. However, the failure of genetic explanations draws attention back to the possible role of family factors. And indeed, we've a couple of surveys of clinicians in Australia and elsewhere, which shows that quite a few clinicians still cling on to the idea that families cause eating disorders. So I guess one of the challenges that faces is that understanding the role of genetics and how genes work is really complex. And so it's not that easy to understand, and hopefully what we're going to achieve today is to make it all a bit more understandable. So there's a couple of major problems with the family hypothesis. The first is that whilst cross-sectional research does consistently show that as the severity of eating pathology increases, so too does the proband reports of family dysfunction. But longitudinal research shows that family environment can only be viewed as a retrospective correlate rather than as a true risk factor for the development of an eating disorder. And this is based on the excellent work of Corinna Jacobi and her summary of the various longitudinal studies that have been conducted in the field. There are only 2 studies to date that show exceptions to this lack of association. There is a finding that low levels of social support from families are associated with the development of eating disorders, and abusive parental relationships are also associated with the development of eating disorders. But what we have is numerous twin studies, and when there's been a summary done by Kelly Klump and her group, shows that genetic influences account for around 52% of the variance that contributes to eating disorder etiology. So we're not talking about a minor contribution, and that is a median figure, so we're not suggesting that that is the same number for everybody, but it's certainly a major contributor to the reason why eating disorders emerge. Now, what is it about twins that helps us to understand what it is that's contributing to a disorder? I've got a slightly fuzzy picture here, but as you can see, it's the twin model, and we're looking at twin 1 and twin 2, and we're looking at what we call latent sources of contribution. The A stands for additive genetic, the genes that contribute to the emergence of a trait, in this case the eating disorder. C stands for shared environment. That's the environment that both twins experience equally. Now, that could include family variables, but it could equally include sociocultural or peer values or peer experiences. E stands for the non-shared environment, the environment that is unique to each twin in the family. So an experience that happens to one twin but not to the other, or even an experience that they interpret differently from the other twin. So the beauty of twins is that, of course, we have a little bit of a living laboratory in that we've got identical and non-identical twins. Identical twins share 100% of their genetic material. Dizygotic or non-identical twins share on average 50% of their genetic material, the same as any sibling in any family. So what we use are basically mathematical models using that understanding to find out the variance of genes, the shared environment, and the non-shared environment. Now, when we do that, and this is summarizing quite a few studies, you'll see in that second column, and this is looking at anorexia nervosa, bulimia nervosa, objective binge eating, vomiting, importance of weight and shape, intentional weight loss, and disordered eating, often measured using the Global Eating Disorder Examination. So you can see across these studies, the take-home message here is that both genes and non-shared environment are important. Particularly, genes and the non-shared environment show a significant contribution to all of these phenotypes. One thing that sometimes worries people is there's quite a broad range of estimate. So you can see for anorexia nervosa, the estimates of genetic variability range from 31% to 76%, 28 to 83% for bulimia nervosa, and so you can see that the variation is with the non-shared environment. Now, this really probably reflects the quality of our measures. So if we have measures that are not so accurate, that may mean that we have a higher estimate of the non-shared environment because we've got more measurement error. We also know that if we have more valid interviews, we usually get a lower estimate of the genetic variance. So a few things to take away from this table. One is that the only thing that doesn't convincingly have a contribution of genes is the importance of weight and shape. That's that diagnostic criteria that is present for both anorexia nervosa and bulimia nervosa, which is the degree to which people judge their self-worth in terms of their weight and shape. When we step back and look at weight and shape concern, which is a slightly broader concept and also includes body and shape dissatisfaction, that does have a significant genetic component. Okay, well, what about the shared environment? I haven't really talked about that yet. And doesn't come up very often. It's been detected in 1 out of 7 studies for bulimia nervosa, 1 out of 17 for the importance of weight and shape. It is more commonly found, however, in continuous measures of disordered eating, but it usually contributes less than either the genes or the non-shared environment. And it may be found in things like for example, the global EDE measure, and another very strong finding that started first with Kelly Klump's group is that the contribution of the shared environment decreases over development, particularly from childhood into adolescence. And we've also conducted a couple of studies, one on weight and shape concern and one on the Global Eating Disorder Examination, and we show the same trend that the shared environment decreases as people enter puberty and the contribution of genes increases over that time. So there is certainly a suggestion that the shared environment does contribute. It's not a major contributor and it's not a consistent contributor. But what are we talking about when we're talking about the shared environment? Well, this table is from my research and looking at weight and shape concern over adolescence. We had almost 600 female twins. We've followed them up over 3 periods over adolescence. And you can see here we've divided the data into the ages 12 to 13. We then followed them up again when they were 13 to 15, and we followed them up again when they were 14 to 16. Now, up the top there, you can see what I was talking about. The contribution of genes is 1% at 12 to 13 years. It increases to 17% at 13 to 15 years, and up to 22% at 14 to 16 years. Remember, we're talking about weight and shape concern here, not behaviours, which typically have a higher genetic contribution. The shared environment, as I suggested, starts off a bit higher at that younger end, 34%, goes down to 32%, and then at 26% at ages 14 to 16. The non-shared environment has the lion's share of the variance here, 65%, 51 and 52%. Now, if you look down each column, you'll see that there's a number of highlighted red cells, and these are significant associates with weight and shape concern at that age group. In the left-hand column, you can see the variables that we're talking about. So we measured a variety of parental measures: parental criticism, parental expectations, parental conflict, maternal care, paternal care. And we also looked at peer teasing about weight and appearance, media internalization, the pressure to be thin, the pressure they received often from peers or the media, and adverse life events. Now you can see at ages 12 to 13 that a lot of the parental variables are not significant contributors. And when you look at the numbers in the brackets, they're the effect sizes. And you can see by a long way, the strongest effect sizes at 12 to 13 years belong to peer teasing about weight, media internalization, and pressure to be thin. And if you look over to the other two age groups as the twins get older, 13 to 15 years, it's a very similar story. You can see that the effect sizes associated with the family type variables is certainly not as big as those associated with the peer teasing, the media internalization, and the pressure to be thin. So the shared environment, particularly as the girls were reaching 12 to 13 and onwards, is very much seems to be focused in, I guess, the socio-cultural environment that a lot of us share. We are certainly living in a society where the thin ideal is the desired ideal. We get a lot of visual material about what that looks like and a lot of pressure to look like that. So from our research, I would suggest that the shared environment centres probably more on the socio-cultural environment. The other useful thing about twin studies is that they can be used to examine the overlap between genetic risk factors for different disorders with eating disorders. And there's two major areas that have been examined between eating disorders. The first is depression, and the second is drug and alcohol or substance abuse. So this slide here is really looking first of all at the overlap with depression. And the summary here is that the association between the two phenotypes, we know that eating disorders and depression occur quite commonly in our clients, and that is primarily due to shared genetic risk factors rather than an overlap in environmental risk factors. So, for example, the correlations between genetic risk factors for depression with anorexia, the correlation is 0.56. It's 0.46 with bulimia nervosa. It's also been examined with disordered eating and been found to be 0.70. And we also looked at the overlap with any eating disorder, whether it was anorexia nervosa, bulimia nervosa, binge eating disorder, or purging disorder, and suicidality, and that was 0.60. You can see there with the confidence intervals, they all show that these are significant correlations. And so we know that there are some shared, it suggests some shared pathways with depression that relate to biological and genetic pathways. The second area where we've looked at overlap is between bulimic disorders and substance use disorders, and that includes things like alcohol, illicit drugs, caffeine abuse, and dependence. And it shows that the correlation between genetic risk factors is significant and moderate, and it ranges from around 0.35 to 0.53, again with very little overlap in environmental risk factors. So the reason that bulimic disorders and substance use disorders co-occur is primarily due to shared genetic risk. So if we look at some specific studies, we can see that alcohol use and binge eating and compensatory behaviours overlap, alcohol dependence and binge eating overlaps, alcohol use and bulimic behaviours also overlap. So both the binging and the purging. There have also been studies done on the overlap between OCD and anorexia nervosa and generalized anxiety disorder and anorexia nervosa. Both show significant genetic correlations, but would suggest that there is a more meaningful overlap with genetic risk for generalized anxiety disorder than obsessive compulsive disorder, which is interesting because we may have predicted just from what we see in the clinic that OCD was going to have a stronger genetic association with anorexia, but the data to date would suggest that that is generalized anxiety disorder. Okay, so where are we then in terms of the bigger picture? We know now that both genes and environment are important, and this is really a summary of a diagram from one of Corinna Jacobi's reviews, looking at all the different risk factors over the lifespan that contribute to the emergence of an eating disorder. I've highlighted genetics and female gender. We know that females are probably around 10 times more likely to experience an eating disorder. The finding around abuse or neglect, particularly implicating the father, is there, gastrointestinal problems. And then into the teenage years, we're looking at things such as difficulties with emotion regulation, escape avoidance coping, low social support, perfectionism, negative affect, ineffectiveness, weight concerns, and dieting. Now, the genetics and the environment, they're both important, and really important to understand that neither is deterministic. And we still often get the question, it can be from the media, but the question, is it genes or is it the environment? And of course that question doesn't even make sense because we know that it's both, and there is really very few disorders that I can think of that would be caused by either one or the other, particularly any complex psychopathology such as we're looking at with eating disorders are going to have both involved, as the twin studies suggest. Now, taking it a step further, we have to get our head around the dynamic interplay between genes and the environment. It's not just that they're both involved, but they're actually quite dynamic. And to try and make this point, I think I found a very helpful in one of the papers that I've read where they've likened it to Harry Potter. So apologies to those of you who haven't read Harry Potter and have got no idea. There's probably only 2 of you in the world. But this is a quote from one of the books. Harry unwrapped his chocolate frog and picked up the card. It showed a man's face. He wore half-moon glasses, had a long crooked nose and flowing silver hair, beard, and moustache. Underneath the picture was the name Albus Dumbledore. Harry saw, to his astonishment, that Dumbledore's face had disappeared. "He's gone." "Well, you can't expect him to hang around all day," said Ron. "He'll be back." "But in, the Muggle world, people just stay put in photos." "Do they? What, they don't move at all?" Ron sounded amazed. "Weird." So I think that's a good simile for what we're looking at here. We've often thought that genes and environment, I think we've got to the point where we understand both are involved, but often we sort of have had a sense historically that they're fixed. Once that has a person's immersion to the world, there's not much that can be done. What we've been finding, particularly over the last 10 or 15 years, is that this is not the case, that in fact both the genes and the environment are moving around, contributing different variances at different times of development. So gene and environmental influences are not static, like subjects in Muggle photos. Instead, they are like subjects in wizard photos. They transact or interplay with each other, sometimes strongly present, other times asleep or even absent. So when we talk about gene-environment interplay, there are four main mechanisms that we're talking about. I'll go on to talk about each of these. The first is variations in genetic influence according to environmental circumstances. So this is a quantitative relationship. The second is epigenetic programming, where the environment affects gene expression. There are gene-environment correlations and interactions between gene and environment, between specific DNA sequences and specific measured environments. So let's look at the first one here, variations in genetic influence. Now, just a general example here is the income health gradient. This is the robust association that we see all around the world between income and physical health. Basically, people with greater financial resources tend to have better physical health. Now, this is in part because they can pay for better healthcare, but we also know that income influences biological functions that in turn affect health status. So it might be that there is less stress in the environment, and this in turn we know has a biological effect which affects health status. Epigenetic program is something that, if you're wanting to read more about, Howard Steiger has just written a good review on this and its relevance to eating disorders. So to try and understand this, we're talking about heritable changes in gene expression, and this is active versus inactive genes that don't involve changes to the underlying DNA sequence, but it's a change in the phenotype without a change in the genotype. And the diagram there gives examples of the different types of environment that might influence the underlying, the genetic expression. You've got diet, you've got seasonal correlations, disease exposure, exposure to toxic chemicals or drugs, financial status, exercise, the microbiome, therapeutic drugs, alternative medicines, social interactions, psychological state. So if we go back to our monozygotic twins, remembering that they both have the same underlying DNA sequence, but in the womb, they can start to experience different environments, which may actually mean that one monozygotic twin has some particular genetic expression which is being turned off, whereas the other one remains active. An example of this, which doesn't involve twins, is the Hongerwinter. The Hongerwinter in the Netherlands, 1944 to 1945. It was the end of the Second World War. and there was a German blockade which caused starvation amongst the people living in the Netherlands. So people were starving, and the children conceived during this time experienced persistent detrimental health effects 6 decades later, including diseases like diabetes, obesity, and cardiovascular disease. And that was associated with less DNA methylation of the imprinted IGF2 gene. So essentially, it appears that the starvation impacted on genetic expression, and that then led to an increase in disease. The interesting thing about epigenetic programming is we think that that changed genetic expression is heritable. In other words, it's handed down to the children. So this changed genetic expression was also present in the children of the children born in the Hunger winter. Now, in eating disorders, we have a number of credible epigenetic triggers. We don't know for certain, but here are some ideas. The first is preterm birth and all the, I guess, the assaults that can come through that in terms of impact on the baby, like some sort of birth trauma. Season of birth, sexual abuse, adverse life events. If you look at the work that Howard Steiger's done, he's particularly interested in early stressful life events that can impact on the expression of genes. And interestingly, change in dietary intake. Now that may be restriction or overconsumption, but we know often with our clients that a change to their dietary intake, particularly restriction, can be the start of a catastrophic move into an eating disorder. But not everyone who goes on a diet experiences that. So hence it might be that dietary intake is triggering some change in genetic expression in some people but not others. Puberty, of course, is another example of some sort of epigenetic trigger that may be relevant to eating disorders. Looking at gene-environment correlations now, these describe the extent to which individuals are exposed to certain environments as a function of their genetic vulnerabilities. So there are three types here that we talk about: passive, evocative, and active. Passive gene-environment correlations are where parents provide their children with both the genes and an environment that is conducive to the development of a certain trait that will occur independently of the offspring's characteristics. Now, an example here is we know that mothers who have eating disorders are significantly more likely to have young children who have feeding problems. And this is in part genetic. It's in part environmental. In terms of videotape meal times, we know that mothers with eating disorders who are often quite stressed and quite uncomfortable with handling food often use more verbal control with their children, more likely to express negative emotion, and to be less facilitating with exploration around food and a bit more intrusive in terms of managing food. So often if children can't explore with food, and that includes throwing it on the floor and smearing it on their face and all the things that we find so endearing, if the environment's controlled too much, then they often can result in a feeding problem or refusal to eat. So we can see here that that's where both the genes and an environment is working towards the same end. An active gene-environment correlation is where individuals actively select or create environments that are correlated with their genetic propensities. So here we have, for an example, a co-twin control design. This is a recent study with female twins aged 8 to 14 years. And it showed associations between disordered eating and weight-conscious peer group association. So we know that girls with disordered eating are often hanging around with peer groups who are talking a lot about weight, talking a lot about appearance, comparing different diets. When we look at the reasons for this using the twin design, they found that these associations were largely due to genetic and shared environment selection factors rather than pure socialization effects. So genetically, people are selecting out peers with similar interests or similar concerns. Evocative gene-environment correlations occur when genetically influenced characteristics in the child evoke an environmental response that reflects the genetic trait. So I've given an example here. I've made this one up, but credible in terms of, again, what we see in our clients, where a harm-avoidant, timid, anxious child can evoke a response of overprotection from concerned parents. Now, we know that that happens in children with anxiety disorders, and where children who express anxiety about going out and socializing, parents, out of concern, will say, well, don't push yourself, don't go out, therefore preventing exposure. Now, this in turn can lead the child to become more timid and uncertain of their autonomy, and may then place them at risk of developing an eating disorder. So they're evoking concern and anxiety from others in the environment, reinforcing behavior which ultimately places them at risk. Okay, the fourth aspect of gene-environment interplay was the gene-environment interactions, and this is probably where we see most of the research at the moment, and this shows the effect of the environment depends upon the genotype, such that individuals will be varyingly susceptible to the influence of high-risk environments proportional to the degree of genetic risk. So this goes some way to explaining why the Western socio-cultural environment impacts adversely on some people but not others. If it was a uniformly adverse influence, then we would all be developing eating disorders. But maybe it works because it triggers eating disorders in those who are genetically susceptible to developing an eating disorder. So what's interesting about this is if we can identify the environments which trigger problems in those who are more genetically vulnerable, then to me this would suggest these are the environments that we prioritize in terms of interventions to try and protect people. So again, pulling on the work from Howard Steiger, this is an example of a specific gene and environment. He's looked at the serotonin transporter gene, promoter polymorphism, 5-HTT-LPR. You would have probably heard of this. It gets mentioned often in the psychiatric literature. The work of Caspi first directed our attention to this, looking at this polymorphism and its interaction with stressful life events and leading to depression in young people. It's certainly associated with affective instability and behavioral impulsivity, and the polymorphism in interaction with childhood abuse was associated with significant proportion of variance in stimulus seeking, insecure attachments, and borderline personality disorder in a population with bulimia nervosa. So it doesn't directly lead to an eating disorder, and this, I guess, is helping us to embrace the complexity of these gene-environment interactions, but it certainly contributes to a temperament and affect, which then places people at risk if it's also in the presence of childhood abuse. Now, I've summarized here on the table a number of studies and that have looked at gene-environment interactions. Some of them have looked at specific genes. Some of them have used twin studies and modeled a latent genetic risk. And you can see the majority of the studies look at early stress. And so we've got attachment experiences, childhood trauma, adverse life events, parental divorce. So a lot of focus on early stress and how that might make those people who are genetically vulnerable for disordered eating be likely to develop an eating disorder. The last three on that list look at puberty and dietary restraint, contributing to disordered eating, and dietary restraint to the emergence of binge eating. We can add another study to that list, and I'll be talking about that a little bit later, but we've just had a paper accepted in press now, which uses twins to model genes with peer teasing about appearance, which increases risk for disordered eating. So we'll be looking at that in a little bit. The thing that I do want to stress with gene-environment interactions is this idea about the orchid and the dandelion. And so if you look at the graph there on the right-hand side, you can see the people with the X genotype, if they have that and the environment is difficult, then problem severity increases. But you can also see that as the environment decreases in intensity, if there's actually a protective environment, the problem severity decreases. And this compares to the people with the Y genotype who are not genetically susceptible to disordered eating. So adverse environment or a positive environment doesn't particularly make much difference. They're known as the daisies, the hardy ones that don't really get to the highs or lows. The orchids, they flourish in a favourable environment, but they do poorly in an adverse environment. So for all of the people that are genetically susceptible to an eating disorder, in the right environment, they can actually flourish really well. And I think, again, for those of you who are clinicians, we can see that in our clients. As they learn to manage their environment, they go from something that's been quite catastrophic and quite destructive. And as they move into recovery, they start to harness strengths and really move along and start to do really well. So it's sort of like the flip side of the coin. And I think, again, it just shows the importance for us as people involved in this area of providing or looking to enhance helpful environments in our interventions, whether we're talking about prevention or whether we're talking about treatment. Now, we don't know who our orchids are. There are some proxy indicators. It might be people with high weight and shape concern, high drive for thinness, high levels of perfectionism or driven exercise, or negative affect. But we know that certainly those are the people that are going to have that genetic susceptibility to disordered eating. And another example, I guess, clinically, is we often hear about clients, it's that one occurrence, that one life event that pushed them very definitely into the eating disorder. And so it might have interacted with the need for perfectionism and control of their environment and really move them into an eating disorder. Okay, now I've been talking a lot about genes. And you'll notice that I haven't particularly talked about any specific genes. And that's because we don't know. As we said, as I used that reading at the beginning of the talk, we don't know which specific genes are associated with eating disorders. Well, why is that? Is it looking for a needle in a haystack? Are we deluding ourselves? The answer is no. The reason is very simple. In order to start locating specific genes, you need a lot of participants, a lot of blood samples. You need a big number, bigger numbers than we ever realized. I've got an example here from schizophrenia to try and make this point. You can see in 2009, with 3,000 cases, there wasn't much happening in the way of identifying loci for schizophrenia. 2 years later, in 2011, they had collected 9,000 cases, and with blood samples looking at the genes, and at this point, 5 loci were identified. So 3 times the cases, we're starting to pick up specific loci. Now in 2012, just 1 year later, we now have 14,000 cases, 22 loci have been identified. So it's really starting to pay off, getting more numbers. And in 2013, 31,000 cases, 78 loci. So it's essentially an issue of power, that you need a lot of cases to start to identify the loci, and the experience across different disorders, whether they're psychiatric or physical, has been that the more the cases, the more the loci. And eating disorders are not going to be any different. So in 2014, with schizophrenia, with 38,000 cases, they're up to 128 loci. So we have not quite reached that tremendous effort in eating disorders, but efforts are indeed underway to increase our sample size. And this initiative is mainly led by Cynthia Bulick in the United States. And the particular part that I'm involved in, very low down the food chain, but involved nevertheless, is the ANGI study, the Anorexia Nervosa Genetics Initiative. And it's an initiative of the Klarman Family Foundation, and it's across 4 different countries. And we're starting to collect quite a large sample of people who have had or have anorexia nervosa, and comparing those blood samples to unaffected people. And I would say, from what I'm hearing from Cindy, that in the next 2 years we're going to have some very specific findings starting to emerge for anorexia nervosa. So I think in 2 years' time, if I was to do this talk, there would be quite a lot to be reporting in this space. We haven't yet started this work really with bulimia nervosa, but the principle was to start with anorexia nervosa because it's considered to be a more homogeneous disorder, and therefore will take a bit less power to identify the genes. Okay, you say to me, well, identifying the genes for eating disorders, why does it matter? What difference is it going to make to us on the ground? Well, I don't think any of us would disagree that we need more effective treatments for eating disorders, and new genes discovered for other complex traits are already elucidating causal biological pathways. This will give leads for new therapies, whether they be drugs or interventions. So we're seeing this in macular degeneration and asthma. So an example with asthma here, the quote, "This is an exciting time in the field of asthma and allergy genetics, as a list of well-validated susceptibility genes are emerging and defining important biological pathways." In particular, the accumulation of genetic studies exploded assumptions and showed that genes in non-allergic, non-immune pathways may have important roles in asthma pathogenesis. So really the take-home message is that by discovering the genes, it can help us think outside the box. We all get captured in our particular theories and hypotheses about what might be happening, but discovery of the genes and the biological pathways that they imply can really help us step outside of our usual thinking and start to see the novel sorts of pathways and the diverse pathways that may be contributing to disorders. So understanding more about genetics will also help us target prevention strategies more strategically and efficiently, including universal prevention, which is an area close to my heart. I'm just going to give a few examples and about how this can work with the studies that we've done to date. First of all, understanding the key developmental periods will point us in the direction of when is it most critical, or when will it give us most bang for our buck to intervene. So this is a study we did of weight and shape concern. This is with the twins that I was talking about before as they get older. So you've seen some of this data. This is just a different way of presenting it. The graph shows you the green is the genes that are contributing to weight and shape concern as they mature. The red is the shared environment. The yellow is the unique environment. And we've talked already about the changing contributions as they age. And this would suggest to me that getting in at about age 13, which in Australia is when they start high school, but getting in at 12 to 13 age, just before there's an increase in genetic contribution, to try and protect them against those environments that are most likely to interact with that genetic susceptibility, might be a really key time to offer an intervention. This is also from our research, and it's the same group, but what we did was we followed them through now to an older age, 16 to 19, and compared it to when they were 12 to 15. Now, what you can see, we've got this is the disordered eating, it's the global eating disorder examination, and you can see at ages 12 to 15, we've got the contribution of genes in the blue, shared environment red, non-shared in the green. And the asterisk at the end indicates that the non-shared environment was a significant contributor at ages 12 to 15. When we looked at them again at ages 16 to 19, you can see those sources of genes and environment are still contributing, particularly significant contribution of the non-shared environment. But we now have new significant contributors, a new source of genetic variants and new sources of non-shared environmental variants. And so, in other words, that would suggest to me that ages 16 to 19 are another critical time for intervention. There seems to be another wave of new risk factors, both genetic and environment. And this sort of accords with what we all know, that at early puberty and then at late adolescence are two really tricky risk periods for the emergence of disordered eating. So this maps nicely onto that and starts to suggest that they're both important age groups to be looking at prevention work with. We also look at modifying reactions to stress if those who are genetically susceptible, if stress is going to be difficult for this group. And so whether mindfulness is something that can be useful. I'm just, again, picking up examples, thinking outside the box here. And this is from our work published in 2015, where we looked at teenage girls. They were aged around 16 years of age. And we gave them, there was a control group that received a mindfulness intervention for 3 lessons and a group that received a cognitive dissonance intervention. And you can see there, in terms of eating disorder symptoms over the 6-month follow-up, the control group did what we'd expect them to do, which is slightly increase. The dissonance decreased a bit, but the mindfulness had a significant decrease compared to the control group. The Information about genes and environment can also help us identify key environments that increase genetic risk. So this is the gene-environment interactions. This is the puberty study that I was talking about before. If we had no gene-environment interactions, you can see what we would expect to see on the left-hand graph. But what we actually found is in the right-hand graph that there is, in fact, an increase in genetic risk as peer teasing increases. There's also an increase in non-shared environment, but it's not as steep as the genetic increase. So peer teasing appears to be a particularly toxic, excuse me, particularly toxic environment. And we do have interventions that can decrease peer teasing, and we certainly have some success with these interventions in decreasing the risk factors for eating disorders, or actually decreasing the development of disordered eating, as with the Body Project. The epigenetics of dietary restriction are also interesting. Dietary restriction is seen as a really important environmental trigger of genetic action across a whole range of disorders, and it's likely to be a powerful epigenetic trigger in eating disorders as well. And that does remind us of the importance of integrating prevention approaches with obesity to ensure no harm is done. As we would know, the main message with obesity is often about watching what we eat and being careful with what we eat. But if that message is given to genetically susceptible people for eating disorders, then that could certainly be harmful. So we need to think about responsible integration. Also, the information that I presented about genetic overlap with depression and substance abuse, that may suggest that borrowing approaches from these fields to inoculate youth may be effective. So, for example, there's a lot of work being done on attention and interpretation biases related to negative mood, and we've done less of that in eating disorders, but it might indicate that that would be a fruitful area of inquiry. We also know that perfectionism underlines a lot of negative affect, including depression and anxiety, and of course is associated with eating disorders. And this is an earlier study of ours which looks at 3 groups treated for bulimia nervosa, a waitlist no treatment, it was a waitlist no treatment. They didn't change much over time, as you'd expect without treatment. People that received a perfection intervention, what I've got listed there are the effect size changes. You can see a large decrease in disordered eating, large increase in self-esteem, large decreases in depression, anxiety, and perfectionism. And compared to the CBT at baseline, then they had a large impact on disordered eating, but not so much on the other variables. So it might be that perfectionism is a particularly interesting area of inquiry. And again, we know that that's something that can be prevented. You can see in this study that we published in 2015 that we were able to significantly decrease perfectionism in year 8 to 10 boys and girls over a 12-month follow-up period. And at 6 months, that was associated with significantly lower levels of self-criticism and negative affect. So if we're thinking outside the box, this offers opportunities to start connecting the dots. So in summary, there is a substantial contribution of genes and non-shared environment to all aspects of disordered eating. We're less certain about the impact of the shared environment. We know that there is certainly some contribution to some phenotypes. We're only really starting to examine the dynamic interplay between genes and the environment in eating disorders. And so very much at the beginning of that, as we are at the beginning of starting to identify the specific genes that contribute to eating disorders. But we need to really use this developing understanding of this dynamic interplay to help us think outside the box of developing interventions. We always need to complete the loop. Understanding about the genes and the environment is not enough. We need to think about how that informs our interventions and allow that to lead us into new areas that can benefit people with eating disorders. So I think, yep, that's the end of my presentation, and I'm now going to hand it over to Emily.
Wonderful. Thank you so much, Tracy, for the presentation. And it was great to hear about the current data on genetics and eating disorders and to see how the research is progressing. And I think I can speak on behalf of many of the listeners today that this is an exciting area, and I'm very much looking forward to seeing what unfolds when we reach the necessary power with the data. We don't appear to have any questions at the moment, so I would firstly like to take this opportunity to thank all the members for taking the opportunity to listen today, and that the presentation will actually be up within the next 24 hours. And should you have any other questions that you'd like to address to Tracy, by all means contact Etta Carter at the AED for any additional information. So thank you again, Tracy.
Thank you.
Okay, it actually appears we do have a presentation, a question about the presentation. So a question at the moment is for you, Tracy. What would your top 3 interventions for prevention be?
Well, I guess it depends very much on the age group and the setting. So I think one thing we have learnt is that an intervention might work better for older people, older adolescents, but doesn't work well for younger. I guess we've, from our own research, showed that with Media Smart, which is a media literacy approach that we use with 12-year-olds, 13-year-olds, when we looked at it with 15-year-olds, it didn't seem to have much effect. And we sort of figured by that stage people had got so immersed in the media that they were too far gone in that sense to be inoculated against those messages. In terms of what we know seems to yield the strongest results is certainly the cognitive dissonance approach, and that has been used with really most of the studies have looked at older adolescents or young adults who have concerns about their body image. So they're starting to move towards being at risk. And so the data is certainly substantial in that area to show that that does decrease risk and has indeed decreased emergence of eating disorders compared to control groups. So I think with the younger people, I would direct people's attention to a meta-analysis that is in press in International Journal of Eating Disorders, and Hannah Watson is the first author. And it's a meta-analysis of prevention studies for universal prevention and selective and indicated, which basically means universals when it's given to everybody regardless of risk, selective and indicated when it's given to people that are starting to move into the risk spectrum, like girls with body image problems. They showed with their meta-analysis that media literacy approaches were showing the most promise with younger universal audiences, and so I would encourage people to look up that meta-analysis. And it also looks at cognitive behavior therapy approaches and the cognitive dissonance approaches. So I think that actually provides a very nice summary of our knowledge to date, and also I think gives us some ideas for where we need to be heading in the future. We still have a lot of improvement to go, and as you would have seen by the slides that I put up, I like to to experiment with different things and look at different things to see how far that will take us. And I think we do need more of that to investigate.
Fantastic. We actually have another question. This is from Melanie Rogers with regards to perfectionism. And she asked, what was the intervention you used to treat perfectionism that had such a positive impact in teens?
We based the intervention that we developed on the Overcoming Perfectionism book that I've written with Ross Shafran and Sarah Egan. So basically, we were. It is published as an outline of, but this, in the study there's a table which summarises it, but we particularly look at the fact we try and differentiate perfectionism from achieving. And I'm always very clear with people about that, because I would never ask people to do things that I wouldn't want to do, and I don't want to be second rate and aiming low. And we've emphasised that wanting to achieve, wanting to strive, wanting to work hard is absolutely fine, but that's different from perfectionism, which is very harmful, which is basically setting higher and higher goals in terms of musts and shoulds and beating oneself up when one doesn't achieve the goal. So we do, in that intervention, talk about self-compassion and practicing self-compassion more in the Paul Gilbert sense of seeing self-compassion as a functional thing that we need to do in order to get back on the bike so that we can carry on with striving, achieving, and being the people that we want to be. So it also looks at the value of making mistakes. And we've actually been doing this work now with primary school children, and we've shown just in 3 lessons with using those sorts of principles in the intervention at 3-month follow-up, we've shown significant decreases in perfectionism and significant increases in self-compassion and well-being. So I think what we try and do with all of our interventions is have a lot of fun, and they're very interactive as well. So we really use a lot of role players and exercises in the classroom, and we have a bit of fun about not being perfect.
Mm. And this is a good question from Tabitha Hume, who is a registered dietitian. And she'd like to ask you, Tracy, what research do you have on epigenetic impact of restriction leading to eating disorders?
There's not a lot of research there. More of the research that I'm aware of around dietary restriction is looking at physical health problems. So I know my epigenetic colleagues have been looking at it across other sorts of areas. So at the moment, the person that does most of the work in the epigenetic field is Howard Steiger's group, and that's been around the stress, early life stress. And so that's an area that we need to be developing more knowledge in. And I am hoping that as we find more of the specific genes, that may well implicate pathways to do with nutrition.
Excellent. And Ashley Smith would just like to confirm the name of the book that discusses perfectionism.
Oh yes, sorry, it's Overcoming Perfectionism, and it is part of the Overcoming series. They have a self-help series which ranges from everything from depression to self-esteem. So it's part of that series.
Mm-hmm. Fantastic. Well, that looks like they're all the questions that we have for this evening or today if you're on the other side of the globe. But again, I'd like to thank Tracy for a fantastic presentation and putting what can be quite a complicated topic, making it as simple as it can be. So thank you so much, and thank you for all the wonderful questions. And as previously mentioned, the presentation will be up in the next 24 hours on the AED website. So thank you.